This presentation is an introduction into the organic compounds proteins and nucleic acids. The building blocks of all proteins are amino acids, and there are about 20 different amino acids in the body, but they all have the same four basic components: a central carbon, a carboxyl group, an amine group, and an R group. The amine group contains nitrogen, and the R group is the part that differs in the 20 amino acids. So this is roughly what it looks like. We have a central carbon, the amine group, a carboxyl group, and then the R group. Small proteins are called peptides, and the bond between amino acids is called a peptide bond. To join the two amino acids together, the process of dehydration synthesis is used. A polypeptide is a large chain of amino acids. Proteins have four different levels of organization. The primary structure is just the order of the amino acids in a protein chain. Since there are about 20 amino acids, I think of them like the letters of the alphabet. And so the primary structure are the words that are formed by these 20 letters. The secondary structure is due to the hydrogen bonds between the amino acids. It could be in an alpha helix or a pleated sheet. The tertiary structure of a protein is the final 3D shape. This structure results from the interaction of the protein chain with the surrounding water molecules. This picture shows a protein that will form part of hemoglobin. Some complex proteins also have a quaternary structure. This is when two or more protein chains interact with each other. These can be either globular or fibrous proteins. Notice here that hemoglobin is more rounded in structure, and there are two blue and two purple protein chains, and that collagen is a long, straight molecule. And so because it's rounded, it's called a globular protein. Because this is long and straight, it's called a fibrous protein. Under certain conditions, the tertiary and quaternary structure of a protein can be interrupted. This is called denaturation, or we would say the protein is denatured. This is a permanent disruption and can be the result of high temperature or an acidic environment. An example of this would be an egg. Imagine you are going to have eggs for breakfast. You take the egg out of the refrigerator and crack it onto the skillet. You have the yellow yolk in the center and the clear white around the outside. As the egg heats up, the egg white becomes white in color and solidifies. Well, if you change your mind and don't want eggs for breakfast, after the egg cools down, it still stays cooked. It doesn't go back to the raw form because the proteins have been permanently changed. A specific type of protein is an enzyme. These are biological catalysts. And recall that a catalyst is anything that will decrease the activation energy of a reaction. When an enzyme is used in a reaction, the enzyme itself is not changed or used up. This means that the enzyme can be used over and over again. When a reaction uses an enzyme, the molecules that need to come together are called substrates. The place on the enzyme that the substrates attach is called the active site. The active site is what gives an enzyme specificity. This refers to the fact that each active site will only interact with one specific substrate. Sometimes this is called a lock and key mechanism. And notice here that we have an enzyme with two active sites for substrate 1 and 2. As these come together, the enzyme itself is not changed, but the two come together. Now we have a product, and the enzyme is still available to be used for the next reaction. Let's introduce the substance ATP. ATP stands for adenosine triphosphate, and this is the energy source that most cells use to complete their work. This is the amino acid adenosine plus 3 individual phosphate groups. The energy is stored within the bonds between phosphate groups, so this is potential energy. When these bonds are broken, energy is released and the cell can do work. So you notice we actually have the amino acid adenine plus a ribose that makes the adenosine, and then triphosphate 1, 2, 3. The fourth type of organic compounds are nucleic acids. You have probably heard of these because DNA stands for deoxyribonucleic acid. Nucleic acids function as the blueprint for protein synthesis. The building blocks of nucleic acids are nucleotides. Each nucleotide has 3 subunits: a sugar, a phosphate group, and a nitrogenous base. So here's the sugar and the phosphate, and usually it forms what's called a backbone. We have sugar, phosphate, and then here is a nitrogenous base. The nitrogenous bases come in 2 varieties, purines and pyrimidines. The purines are adenine and guanine, and the pyrimidines are cytosine, thymine, and uracil. For DNA, the sugar is deoxyribose. The possible nitrogenous bases are adenine, guanine, cytosine, and thymine. DNA has a double helix structure, meaning that it has 2 strands joined together and then twisted. For RNA, the sugar is ribose, and the possible nitrogenous bases are adenine, guanine, cytosine, and uracil. RNA is always single-stranded. To form the double helix of DNA, hydrogen bonds form between the nitrogenous bases from one strand to the other. For the bonds to form properly, the nitrogenous bases need to be in complementary base pairs. For DNA, this means that adenine will always be paired with thymine, and cytosine will always pair with guanine. In the formation of RNA, cytosine will always pair with guanine again, but adenine will pair with uracil. A good follow-up to this introduction to nucleic acids will be provided in another presentation about protein synthesis.